Hi, everyone. Today we present some results from a survey of the ocean microbiomes we did a few years ago. The open question that, or some of the open questions that remain to be addressed, and a new project, a new expedition dedicated to the ocean microbiome. So the ocean microbiome, in essence, it's the ensemble, the invisible majority of the ocean. So it's basically all the invisible organisms, essentially unicellular ones, that live in the ocean, but also includes in the concept their interactions and the interaction with the environment. So we extend the classical definition that is typically dedicated to prokaryotes. What is the problem? So the problem, the difficulty of dealing with understanding the structure and the functioning of the microbiome and also predicting its change in the future comes from the fact that the microbiome is essentially made of three, the microbiome dynamics is made of three complex systems interacting across them. One is the landscape, that is the fluid, the water, with all the complexity of the physics of the oceans, the complexity of the interactions, so the network, but also the complexity of the biology of each single organism. The main point here is that instead of trying to simplify or separate the issues, we try to face the complexity, to assess, to understand the mechanism and to predict. Basically, what we want to do is to put together a different level of organization of the system. That is, a level organization means spatial scales, but also processes. It goes from what happens within the cell, the organism, so biological processes, the interactions, so the network of interaction, which are not only predations or competition, and then all the biochemical processes and the climate, up to the climate processes. It means putting together many disciplines, and in addition to putting together many scales, temporal and time scales. And so you can see this as a nested level of complexity. With this idea, basically what we want to build to really have an understanding of the functioning of the microbiome is to put together these three levels of complexity in a way that is a new conceptual view of the ocean, but also means new sampling strategies to really cope with this nesting level of complexity, and also new tools. So exploring the ocean has been since at least 2 centuries. It's been difficult because the ocean are very vast, and going there with vessels is indeed important, useful, and it has been historically important. But we know that a very limited in space and time sampling from vessels is not necessarily the solution. Now we have satellites, we have a lot of robotics, but the point is, are we really ready for robotics? So another way to pose the question today is, are we ready for a robotic monitoring of the ocean's life? So one element of the recent revolution in the ecology, in marine ecology, has been the introduction of environmental genomics, which by using sequencing, we can now have a new approach to taxonomy. We can have an idea of the evolutionary processes, looking at metagenomes, but also we can have an idea of the metabolic state of the organism in the environment by looking at gene expression, essentially. Considering that, one important step was this cruise, this expedition we organized a few years ago, the Tara Ocean Expedition, and which is essentially a survey of the RNA, DNA of the oceans, of the microbiome. And we basically put together a series of tools that are going from advanced sequencing approaches to imaging, and in addition to standard physics and chemistry. And this allowed us to produce a wealth of data that are now used by the community, which produced as the first main result, I would say, the first description of the diversity, where we found that diversity is indeed significant, is important, but it's also measurable, and so it's limited in a way. We see the limits, and but also it's mostly unknown. So, for instance, of the 150 million genes we have, eukaryotic genes we have described, about 50% is basically unknown. We don't know the function, we don't know even the organism that is bringing them. Another, let's say, main results that I just did a review is that we found that there are many interactions, at least putative interactions, and these interactions seem to be dominated more by cooperation than competition and predation. These interactions can be also then described by subnetworks, so groups that tend to interact more. And with this principle of the modules of the ecosystem, some of us were able to actually have a new view of the carbon pump by basically connecting some of these modules of the ecosystem to the carbon export. Another result we got is to have a sort of first description of the biogeography of the microbiomes by looking at the metagenomes, the similarity between the different metagenomes in different regions of the oceans. And indeed, we found that the biogeographies are dependent on the size, so on the kind of the organism. Even if, let's say, for the smaller one, we basically are close to the biogeography that can be derived by analyzing standard data like chlorophyll data, etc., which suggests that there is a close relationship between the biochemical state or regionalization and the biogeography of these organisms. At larger scale, we actually found that the region provinces are larger and seems to be independent on the environmental conditions. So really suggests that these guys, probably mostly predators, tend not to be too dependent on the prey, so are basically generalists. And so the provinces are basically made by the recirculation of the ocean. So this is a main point, in fact. The main point here, one of the main points today is that really the seascape is not the landscape. So we really have to define new concepts to really better understand the functioning of the system. And this is because we cannot really translate terrestrial concepts to the ocean. So there are gradients at all the scales, from large scale of the ocean, from the size, the scale of the planet down to the scale of the organism. And these gradients are transported by the fluid all the time. So this means that in practice, if you follow a community, and we try to understand the dynamics, and dynamics is always in time, so we want to see how it varies in times the community and try to connect this variability, this variation to processes. We see that in the ocean, the community is going to move at the same time as the time passes. So basically, even if the natural framework for understanding dynamics is time, in the ocean, time implies space in most of the cases. It's just a constraint coming from the specific substrate. And we may even consider that we should really use space-time as our conceptual framework, a little bit as it happened for after the introduction of special theory of relativity by Einstein. This conceptual change is really important, and it has an impact on many issues or concepts. So basically, if I start following a plankton patch in Florida, what I can observe that 5 months after, it is in the middle of the Atlantic and moving fast. And this means also that every kind of process occurring on the population has occurred during the travel by moving. So for instance, an accumulation of predators feeding on this community will occur at the end of the path, not at the beginning of the path. So if I want to understand a coupling between 20 predators, I have to consider also space and not only time. But also it means that the classical concept of niche, that is so sort of subset of values of environmental parameters, is actually has to be extended because of the position in space of the community is not only regulated by the local effect of niche, but could be also just by the transport. So basically, in the end, niches are completely alterated or changed by these processes. So one thing we actually found is that to better understand the biogeography, how it is related to the transport, we basically connected the different stations using a Lagrangian approach to try to understand what is the time to go from one station to another if you travel in the water. And what we have found, in fact, is that as expected, these provinces are flushed down continuously. So it's not a fixed concept. You should really consider it. Water is flushing them out, plankton is going through them, and this is the emergence of a sort of Lagrangian story. So it's a view in time. And it means also that if you want to have provinces of certain size, the plankton crossing them has to have a memory, a long memory, because otherwise it has quick changes. You will not see any emerging pattern. So if you see these emerging patterns, case or the basin, that means you need a memory, a long memory. And in fact, what we found by putting together the similarity between the stations and the travel time, basically that you need about 1 or 2 years as a memory of the system in terms of community. So this was just a quick overview. There are many, many issues that remain open. I would just to underline a few. One of these is that we still have competing conceptual models. So we tend to have, for instance, community models that tend to understand the structure and the relationship between the different species at the community level. And these exercises tend to say that the system is neutral. Basically, it's a stochastic emergence from a stochastic system made of many, many, many species which do not have any specific ecology. There is a more classical approach, which is a niche approach. So basically one trophic level, you focus there, you assume that there is competition for resources, and you try to maybe add in some biology to understand the system as a competition and within niches. And this is actually the approach that it's at the base of our modeling exercise, the current biogeochemical modeling we use. But also more recently, there is this idea that actually we can study interaction by, for instance, looking at co-occurrences. And so there is this idea of networks or structure of the system and interactions. But there is a huge problem because we don't know how to prove these interactions in the ocean. So another implication of the limitation we are currently having in our studies that, for instance, when we do niche modeling to try to predict what is the response of a species to climate change, we are using a very simplified approach using proxies basically. And here, for instance, it means that to predict what is a change in the savanna of the trees, we look only at the relation between the trees and the rain. And maybe we find a very good predictor based on rain data, but in reality, what is impacting the trees is more like animals like elephants. And so when we see this relationship, the rain is actually impacting also the elephants. And so in the future, what is an irrelevant relationship, the direct relationship between rain and trees, or the relationship going through the animals. And it's crucial because if we want really to understand and predict, we need to understand the sensitivity of these different elements to the climate change, not only of the target. Another issue that is quite open is this, as I was mentioning before, is that if you look at community level, recent papers suggest it's really neutral, the community in the ocean, the microbiome community, while at the same time, we see from our transcriptomes, for instance, there is a variability in gene expression. So we still need a conceptual model of the community structure that is coherent with the metabolic processes. And I would add, is actually even difficult if we do not have this conceptual model. It's difficult to interpret the metatranscriptomic data. Another important point is that, okay, now we have access to metabolic state from gene expression in the future from proteomics and metabolomics, but we are not measuring any more rates. But without rates, without the flux of matter and energy, it's very difficult to conclude on a budget, who is doing what in terms of budgets. And that means also that not only it's difficult to understand the functioning, but also it's difficult to derive new models. It can be numerical, could be from artificial intelligence or anything else, but it could be used to predict future responses. Another I don't have time to discuss this, but this is actually an important point, is also the life cycles are never considered in these studies, or almost never considered. But these guys actually have complex life. And but instead, we just consider them as all equal, all adults in a way, in one specific stage of the life, and we do not consider complexity of life cycle. So more generally speaking, we're often able to measure polyproxies with temporal mismatches between the different parameters measurements. Complex system is difficult to sample it because a priori we do not know the variable to measure. So that's why it's really important to have a conceptual model. And also to build a conceptual model is really important to have redesign the sampling to maximize the information we extract from the sampling exercise to really build a conceptual model. Finally, a big issue is data heterogeneity. So genomic data is difficult to compare with, I don't know, imaging, for instance, and also the fact that data are becoming too many. We are soon going to saturate our capacity to sample, to describe extra knowledge. So more in general, to be ready for robotics, which is probably in the future, we need to first bridge the multiple scales and disciplines. We need to integrate lab studies because, for instance, to resolve the problem unknown and create more reference genomes. We need to redesign physical chemical strategies to match the biology, especially in terms of timing. So the gene expression is the response to within a few hours to the environmental conditions. So we have to match the different measurements to really have an understanding of the system. We should redefine operating principles and even redefine indicators of ecosystem health, for instance, and bridge the boundaries between communities, which is not easy at all. We also integrated tools like using the cloud much more than we do now, and basically train a new generation that is able of a researcher that is naturally able to work cross disciplines. One example of future steps is this phenomics. So basically to use to work at a level of single cell or single organism, where you do at the same time a very deep description of morphology together with the description of its DNA and RNA. So in general, we really need to have a circular approach where we connect the computational studies, the laboratories, and going to see in a way that each exercise informs the other in a circular way. And this could happen by, for instance, having process studies and also by augmenting observatories with these new tools, because in observatories you have also labs, so connecting the two, it's really the way forward, I believe. In this context, we have a new European project that just started. I'm the coordinator of it, which is going to be dedicated to the Atlantic Ocean microbiome. So basically, in the next 4 or 5 years, we are going to sample the microbiome in the ocean, and together with other 35 participants from many countries. And within this framework, our pillars are the microbiome, but also the plastisphere, so the microbiome living on the microplastics, and while considering the seascape as a conceptual framework. We will have many activities I don't have time to describe here. There is also modeling, there is a lot of Lagrangian linear modeling, there is also capacity building and forecasting, improvement of forecasts. Quickly, I can say that there will be an increase in our sampling exercise because we are going to have a dedicated flagship going around the Atlantic. We are going to empower coastal sampling in the marine stations. We're going to have a third-party cruises that even if they are dedicated to other exercises, they will bring our protocols on board to try to enrich the database and share with the rest of the world. It is important also here to add that there will be a lot of training and capacity building and literacy, and that we are trying to also to empower infrastructure to really have in this case for Brazil and South Africa, to have to build basically a system of hub and nodes of sequencing centers that they share the same protocols and same approaches to really standardize, because standardize is one of the issues, you know, on this kind of processes. We are now in good contact with the US also and Canada, and so we hope to include them soon. Lastly, quickly, New expedition. With the flagship expedition, this project is a new expedition we are organizing with Tara Ocean, and within this project, within this expedition, we're going to go from this summer for 14 months all around the South Atlantic, doing a series of topic studies, so process studies, which is each study will be has a dedicated team and a dedicated sampling strategy. Each topic study is dedicated to a female scientist, the neighboring country, or of a female scientist who worked in the similar field. We use a state-of-the-art protocols, but we also have a lot of literacy, outreach, and policy activity in the protocols, and we link our activity directly with modeling and socioeconomic studies. In addition to the level of complexity I described before, we are going to add this time this phenomics, so advanced microscopy of single-cell genomics, long-read sequencing, ENA, looking at isotopes, trace metals, but also looking also at metaproteomics and metabolomics at the same time, using metaproteomics with max cycle. So basically, the idea is really to go to define oceanographic entities, so relevant oceanographic processes that are relevant for, that are important for the ecosystem functioning, to go there and to sample in a way that consider this coupling between time and space I was mentioning before. With this idea, so basically, is to sample the microbiome across gradients along currents. This is, I don't have time to describe all the experiments, but basically, I'm going to go through a few important features like the Amazon plume, addressing the role of icebergs in Antarctica, addressing better understanding of whaling in Africa, and also the role of rivers. And another important point, we're going to try to have an experiment dedicated to some mesoscale as a physical process that modulates the biology. Thank you very much, and that's it.